Thanks to Susan for giving us an awesome introduction, and thanks to everybody for being here today. We're excited to have the opportunity to tell you a little bit about what we've been doing at the Cell Science Institute. So today, myself, Melissa Hendershot, and Brock Roberts, Caroline Hookway, and Greg Johnson are going to tell you about what our work has been for the past year or so. So every biologist has seen some variation on this figure, which you all have now seen since Susan previewed it for you in her slides, which shows the various organelles of a cell spatially organized. However, this representation is not a particularly accurate representation of what a cell actually looks like. In fact, we don't really know what all of these organelles would look like in a single cell at all. At the Allen Institute for Cell Science, our goal is to understand the cell as an integrated system. We're initially focusing on the structural elements of the cell, like its organelles. By understanding the structures that underlie the organization of a cell, we plan to be able to predict how these structures actually function. And by looking at cellular organization across thousands of cells, we anticipate that we'll observe many different cell states. For example, we know that cell cycle state dramatically changes cellular organization. We expect that we'll identify many different cellular states by doing imaging at scale, allowing us to probe subtle variations between cells. This work will allow us to understand how structures are organized and coordinated with cell state. Ultimately, we plan to generate predictive models that will allow us to build an atlas of cell organization that's actually based on real data. So our goal is to be able to take structures like these and orient them to known structures in the cell, such as the plasma membrane, defining the outside of the cell, and the nucleus, to actually integrate all of these organelles into a single model and use this information to predict how cells will behave. So as you might guess, the reason that no such picture, like the one that I showed you, exists based on real data is because this is a huge project and a major undertaking. So to facilitate actually making progress on this enormous problem, we have a pretty defined organizational structure within the Allen Institute for Cell Science. So we have a group of people who work really hard on making the cell lines that we actually work with. Then we have the assay development group who figures out how to work with the cells and does a lot of quality control on the lines that we generate. We have a microscopy group whose goal is to actually scale up the assays developed by the assay development team and again produce this kind of high throughput data. And then once we've produced all of this data, we really need a group of people who can tie it all together and create visual representations and predictive models. So today we're going to kind of walk you through what our institute looks like. And we're going to talk about it as though it's this kind of linear progress for a cell line to move through the institute. But in reality, we're a highly, highly, highly connected team. So we're constantly talking to each other and feeding back information between groups, and we're all tied together by the essential activities of our data management team. So we felt like this was a super appropriate context, this team talk format, to talk about what we're doing at the Institute. So before any of this organizational structure existed, the first and most foundational decision that we had to make as an institute was what cells we were actually going to work with. So as Susan alluded to in her introduction, the cell line that we've chosen as our model system is the human iPS cell. And to tell you more about how and why we made that decision and how we work with the cells, I'm going to hand it over to Brock.
Okay, I'm going to use this. Is this working? Okay. Thank you, Melissa. It's great to be here to speak with you. So, as Melissa mentioned, and Susan before her, our endeavor we've chosen to focus our endeavor on the human-induced pluripotent stem cell. And this is a system that we think has a lot of really powerful advantages and reasons to work with. And I just want to walk through a couple of those. So, what is a human-induced pluripotent stem cell? It's human. It's a human cell line. And it's a pluripotent stem cell, and this, let's see if we can get the pointer working. There we go. This refers to the fact that these are actually mimics of the stem cells that exist in the early embryo, in the sense that they can differentiate into a number of different interesting tissues that are important for research in medicine. And have the potential to differentiate into each of these given the appropriate environment. But they're actually induced to become pluripotent, meaning that they're originally derived from a healthy human donor who has donated some of their skin fibroblasts. And this is made possible by advances over the past decade, the ability to induce these cells into a pluripotent state. It's very exciting. They're also very genetically stable. So these cells do not have a propensity, particularly a strong propensity to gain or lose chromosomes, which is actually not true of the majority of cell lines that have been in use over the decades in the field of cell biology. And it has huge advantages for genetic experiments, makes genetic experiments and attempts to manipulate the genome much more interpretable and tractable, which is very important for what we're doing, as we'll hear more about. And finally, these cells are dynamic and proliferative. Here's an example of a human-induced pluripotent stem cell colony in culture. So they grow as flat epithelial sheets. This is a time-lapse. They divide about once a day. They're very active. They do some migrating. So these cells have dynamic processes that make them an important and amenable system to interrogate dynamic cell behavior. But we need a way to look into these cells. What's the time scale? I think this is 3,000 times real time. I don't know actually how long that would be, maybe half a day. We need a way to look inside these cells. And that's where our gene editing endeavor is focused. And I should also say that the work we do, I should also stress that the work we do is in cells that are derived from a healthy donor, fortunately. And we want to understand cells in their sort of baseline state. We want to understand how cells work. But we hope that our efforts can inspire other work in similarly derived cells where the donor may have a disease and where that disease might be learned about and therapies to address it might come about by following this approach. So that's very important. So the process that we use to interrogate these cells and the dynamic processes within them at the single protein level is gene editing. So that's what we mean by gene editing. This is actually a lengthy process. It takes about 6 months to initiate the design on one of these experiments and end up with a finalized cell line. And we're happy that we've actually gone through this process successfully about 5 times now, and we've made available for distribution 5 uniquely edited human-induced pluripotent stem cell lines that are available to any researcher from the Coriell Institute. So we're very proud of this. I also would like to point out before we dive into kind of the particulars of this workflow that we didn't have a template to follow, so we had to do a lot of experiments to get this working, a lot of pilot experiments to optimize each assay. And so we're also proud that we have that. The first thing I want to talk a little bit about is sort of the initial steps of this, where we design experiments harnessing the power of the CRISPR-Cas9 system to introduce DNA damage in particular locations in the genome of these human-induced pluripotent stem cells, and then our efforts to hijack the response the cells have to that DNA damage by including additional sequence. And we want that sequence to be useful in a way that allows us to label and follow the proteins that are produced from that sequence. And this is what we mean by editing. This is what our team defines as editing. So the important thing to understand about the CRISPR-Cas9 system is that it is an enzyme complex that when complexed with an RNA with a particular sequence, an RNA molecule that we can obtain through the mail after ordering up, rather like a primer or something relatively simple like that, this can be actually chosen to specifically pair a short but specific sequence in the genome of the cell into which it's introduced, and DNA damage will occur at exactly that site at a very high rate. So this has become a very well-known system, and it's revolutionized biology over the last few years, and we're fortunate to be able to work with it. So the important thing to understand about this process is that it introduces damage. So the DNA is broken, and this opens up an opportunity for us because we can actually bias the way that the cell responds to that damage. So an example of how we use this system and harness this approach, it would be our efforts to edit by adding sequence to the TUB-A1B gene, and this is the gene that encodes the protein alpha-tubulin. The important thing to stress is that although genes in the human genome are relatively complex in their structure, the sequence in this gene that's responsible for encoding the alpha-tubulin protein is labeled in orange. And this is a gene that's present in 2 copies as are most genes in the human genome that are not on sex chromosomes. So to start this process, we introduce into these cells the Cas9 CRISPR complex, which we have formulated in a test tube with an RNA that is complementary to a sequence relatively toward the beginning of the coding sequence of alpha-tubulin, this orange part. And oftentimes for scientific rigor, we do this with multiple RNA sequences in this general region to accomplish the same thing. What happens is a DNA break occurs at a relatively high frequency in this region. So these DNA molecules in the genome, the chromosomes themselves, become broken. Most of the time, human cells cope with this damage, which is happening all the time environmentally. It's happening in my cells right now. Most of the time, this process is simply reversed and the chromosomes are stuck back together. Sometimes small mutations occur here. This is commonly, a lot of times, when people refer to editing by CRISPR-Cas9, they're referring to the fact that small mutations can occur when this process is reversed via the human cell's most common DNA repair pathway. But actually what we do is something different. We take advantage of a less frequently utilized DNA repair pathway where the cell is actually open to suggestions. So one way the cells, again, less commonly, but one way the cells can cope with this damage is by copying an artificial molecule that we introduce into the cells, an artificial DNA that has exact sequence similarity to the region in which this damage is actually introduced. I think my pointer might be going, there we go. So this artificial DNA that we've introduced into the cells has the same sequence as the region we've damaged, but it includes something additional, which in this case is the sequence encoding the green fluorescent protein. And the result of this process is actually a new copy or allele that's been modified usefully to include the GFP coding sequence in the coding sequence of alpha-tubulin. So the protein that's made from this tagged allele, as we refer to it, will contain this tag. And this is very useful for us. We typically only introduce these types of changes into one copy of the gene that we're trying to edit. And we usually leave an untagged allele. So, as I mentioned a couple times, it's not a highly efficient process to introduce this useful tag into a copy of one of these genes. So we need to introduce a step where we enrich for cells in which the events we've intended to occur indeed have occurred. And we do that with fluorescence-assisted cell sorting. So this is actually very important for our process. This is a machine that can measure the level of GFP fluorescence from these cells. You can see that compared to a sample on the left, a sample on the right here where editing at the TUB-A1B gene has been attempted gives rise to a distinct population of GFP positive cells. And the cell has the ability, this machine has the ability to actually isolate these cells and reject those that are below a certain fluorescence threshold. So we can actually make a new population that we can work with derived exclusively from the cells that are presumptively edited in the manner that we want to include the GFP tag on alpha-tubulin. So we can keep these cells. And it's not highly efficient. We tend to get anywhere from 0.1 to 5% of the initial population edited in this manner so that it shows up on our as having elevated GFP signal. But this is a big deal because human cells aren't highly efficient for this DNA repair process, and we've had success in 18 of our 20 experiments targeting different genes and at least getting to this level where we can isolate distinctly edited tagged cells. And we're very proud of this. This work is not typically done where a large DNA sequence is asked to enter into a sequence in a gene. Usually people make much smaller changes, and this work's not commonly done in stem cells either. So we're proud of our progress here. The next part of this workflow that I want to talk about a little bit is our efforts to screen through a number of the cells that we've isolated out of our selection experiment to ensure that the engineered changes we've intended at the level of the genome are in fact exact and correct, because we're very we want to make sure that we don't introduce any unwanted changes that could alter how the protein that is produced from the altered version of the gene is in fact regulated and may function. So the way we do this is we take this population that we've enriched for and we sprinkle them at relatively sparse density onto a culture dish, and a number of colonies will arise from this, and we can assume that each of these colonies is derived clonally from an individual cell, and we isolate those colonies and we place each one into a unique well in a 96-well culture plate, and we let them grow for a while. When they're ready, we split those cells and we take their DNA, and on each member of this plate, each clonally derived culture in this plate, we perform a number of PCR amplifications. So I'm not going to talk a lot about each one of these experiments, but they all tell us something slightly different. I've sort of encoded each of the experiments that each of the different amplifications that we perform in a different color. But we amplify all of these different products derived from the untagged and the tagged allele in each cell, and we sequence them, and we ensure that the sequence of those products is exactly what we intend for if the precise edit that we've made is in fact what occurred in that cell, where we've added GFP, the sequence encoding GFP, and nothing extra, and haven't lost anything. And we do reject a large number of cells at this stage, a large number of clones. And then finally, the set of clones that we accept from our genotyping process, after we've ensured that the editing is precise, and this is typically 5 to 10 clones, we subject to a number of strenuous quality control criteria to ensure that after all of this engineering at the level of the genome, the cell around the genome is still a normal, healthy stem cell that can do all the things that a stem cell should do and is suitable for sharing with the community. So the first thing we do is we keep track of the morphology. We make sure this doesn't change throughout this whole process. Anytime this morphology starts changing, that's a sign of premature differentiation, and we don't want that, so we throw those away. We're also very careful to ensure that although these cells, as we mentioned a moment ago, aren't particularly prone to gaining or losing chromosomes, we want to make sure they haven't done that, because it does happen. That has a profound effect on the biology of the cell. We also do deep sequencing. For those who are familiar with deep sequencing technologies, we basically, and whether you are or not, basically we interrogate all of the expressed sequences in the cell and compare it to reference cell lines to just answer the basic question of whether at a global level that cell is comparable, that cell's biology is comparable to that of a reference cell line. We also do more targeted sequencing of sequences in the genome that are similar to the site that we have directed the Cas9 enzyme to for that particular edit. And we want to make sure that the enzyme complex hasn't introduced off-target mutations. And we haven't found any of those yet. And then finally, we also determine whether these cells are expressing protein markers that are correlated with their retention of pluripotency. And there are a number of these markers that are known. Some of them are on the surface of the cell, some of them are in the nucleus. We stain them with antibodies, and we quantitatively measure their level of expression. And the last thing we do is ensure that as a pluripotent cell, these cells are capable of acquiring other fates, and we're particularly interested in cardiomyocytes, as Susan mentioned a moment ago. And indeed, this is an example of a culture where after 20 days in the appropriate chemical environment, these cells have acquired rhythmic contractile properties, and we celebrate the fact that these are robustly differentiating cardiomyocytes, and this is a nice stem cell population that was used to give rise to them. And we do other experiments, but we do these in collaboration with assay development, and most of this work at this point kind of turns over to trying to understand whether the protein that we've tagged is behaving normally and in a predicted manner. So it's a good opportunity for me to turn it over to Carolyn Hookway, who's going to talk more about assay development's work. But I just want to say one more time that we do have lines available. There will be more coming soon. They're at Coriell Institute. And thank you for your attention.
So as Brock, is it on? So as Brock mentioned, we do a lot of work in collaboration between the gene editing team and the assay development team to do a lot of quality control on the cell lines. So even though we can be aware, or the stem cell and gene editing team make sure that the tag is inserted in a very particular location in the genome and nowhere else, we need to make sure that the cells still behave normally even though they have a tag introduced. So the first thing that we do is we look at them on the fluorescent microscope. And so here what you're looking at is a 3D Z-stack of cells that have been edited at the TUBA1B locus. So this is GFP tagged alpha tubulin. And so we make sure that the tagged protein is localized to the structures that we would expect it to be. So in this case, tubulin is localized to microtubules and to the structures that microtubules form. So here we see some mitotic spindles in the image as well. We also do time-lapse imaging of the cells. And so here is a single slice in which we've looked at a time-lapse of GFP tubulin labeled cells. And we can see that these cells are still capable of cell division and that the microtubules are still capable of their normal reorganization during the cell division process. We also do a number of other quality control measures, including transient transfection and fixation and immunolabeling, to make sure that the localization in the edited cells is consistent with the localization in unedited cells. And those things also include Western blotting to make sure that the protein is still expressed in its full length and that the protein levels aren't dramatically changed by the tagging event. And you can see more of that at our poster if you'd like to find out more information about it. But I'm showing you these pictures of epithelial sheets in which there's more than one cell per imaging field. And so understanding where the structure is located in a cell, then, we need to have some kind of reference. So what we use for our frame of reference are the cell membrane and the cell nucleus. So here the cell membrane is labeled in pink, the nucleus is in blue, and tubulin is in white. And the movie should play again in a second. There it goes. So we're going from the bottom of the cells and again to the top of the cells. Now you can tell that there are several cells in this image. But in order to pick out which cells are which, we pick up the relevant information in the images through 3D segmentation. So our team develops algorithms to pick out from each of the image the relevant structures in each channel. So in this case, we have a membrane channel, the edited structure, in this case GFP channel, and then finally the nucleus. So that then we can image across many, many different kinds of cells and look at the variation and the localization of the structures in reference to each other in a way that Greg will describe in further detail.
So once assay development has done a lot of the work to do quality control on the cell lines and also to develop the assays such as the dye assay that Caroline just talked about a minute ago, the cells get handed off to the microscopy team. And our primary goal as the microscopy group is to be able to scale up the assays and cell culture conditions that we learn about from the other teams to be able to do the imaging in extremely high throughput so that we can generate images of thousands of cells. So the system that we've decided to use for doing the majority of our imaging at the moment is the spinning disk microscope. So we have 4 spinning disk setups in our microscopy imaging suite, which is great. And so the reason that we have gone with this particular system is it gives us a nice compromise of being able to take imaging quickly, but still at very high resolution. So this enhances our ability to do things like time-lapse imaging and be able to see how the cells change over time. So in addition, to help facilitate this kind of scale up of the processes that we receive from assay development and the gene editing group, we also have a liquid handling robot that we use to scale up these assays. So we are able to use this robot for both culturing the cells as well as for actually applying the dyes that we use for the imaging. And it's been a really great tool for increasing reproducibility as well as increasing throughput without a poor person having to pipette a million things every day. So one of the things that you can see the robot handling here is something called a 96 well plate, and that is what we use to actually produce the cells that we do imaging on. And one of the things that this facilitates is, again, this kind of high throughput automated imaging. So to do that, we start with a 96 well plate, and we're able to take an overview image of an entire well of a 96 well plate. So we can, of course, do this for every single well in a plate. I'm just going to show you one representative example. So once we have this image showing an overview of an entire well, we can actually segment out and identify where the colonies are. And by using the center of the well as a fiducial marker, we can get the coordinates of each of those colonies in that well so that we can actually drive the automated imaging system, so the objective, to a position in a particular colony that we're interested in imaging. So once we've picked a colony that we want to image, we usually take a slightly lower magnification overview image of the entire colony. So as we've talked about before, the morphologies of these colonies is something that's important to us to understand if they're healthy or not. So we like to have an overview image just showing us the overall morphology. And then we can pick subregions of this colony and actually do higher resolution imaging, again, using these dyes, and then imaging the structure of interest, as Caroline talked about before. So like I said, our goal is to do this for thousands and thousands and thousands of cells. And once we have that data, we're able to hand it off to our animated cell and modeling team, where they're able to utilize this kind of high amount of data to build really sophisticated models. And so I'm going to hand it off to Greg, who's going to tell you about the work that he's doing with that.
Let's see, do I have a laser? Okay, great. You guys can hear me fine. All right, so far the team has talked a lot about how we go from these induced pluripotent stem cells to gene editing, various validation and quality control steps, all the way through image acquisition and scaling. And as the result of that pipeline up to this point are these beautiful images of individually segmented cells. So each object in this field is a segmented cell from one of 7 cell lines. And in each cell, the blue is the nuclear channel, the magenta is the cell membrane channel, and the green is the labeled structure of interest. And you can see that there is a large amount of variation in the localization of each of these structures across the cell lines. But even within one cell line, there's a large amount of variation in texture, in localization. It would also be in intensity if these individual images were not contrast stretched. And one of the things, as we mentioned before, one of the things that we want to do is build sort of an atlas or a model of organization of all of these structures inside of a cell. So how can we get from all of these images to a statistical or a probabilistic model of a cell that allows us to generate images like this? And so with that in mind, we have a sort of a multi-tiered modeling approach. So sort of on the first level is taking the raw images, so without any segmentation, and computing features to allow us to determine, for example, whether or not the colonies are healthy. By the inclusion of reference markers, such as the cell and nuclear shape, we can segment out the individual cells and do sort of image cytometry. So we can measure the size of the nucleus, the membrane, and say things about how the cell populations vary under different conditions, or how cells vary within a single population. Using slightly more sophisticated techniques, we can use rigid and non-rigid registration tools, sort of like what various brain atlases have used to align MRI scans of brains. We use the same sort of technology to align images of our cells to a single sort of coordinate framework to get an idea of where these structures localize. And as a result, we can use these tools to spatially integrate our images of the cells. And sort of on the more sophisticated end, we can use statistical learning models to build conditional models of this structural localization, which I will tell you a little about now. But with all of these tools, we're sort of trying to answer questions about how the cell is organized. So is there a master organizational structure or conditions that force the cell to be organized in a particular way? What are sort of hierarchies of spatial organization of structures inside of the cell, as well as what structures drive reorganization and which structures allow us to predict the localization of other structures inside of the cell. Ultimately, what we'd like to do is be able to build a model of an integrated cell where you could do something like the following. So let's say I have an image of a nucleus here in blue and the membrane in red, and I say, okay, given this cell, I would like to know the position of a particular organelle, let's say mitochondria. And so you take this image, and you say, you tell a computer, you give it to a computer, and you say, I want mitochondria, and the computer thinks about all of the images that it's ever seen before of cells that look like this, and all the images of mitochondria, and it thinks, okay, so what's the probability of mitochondria given this cell and this nucleus and this particular experiment? And then the model returns this image. So this is an example of a real image, but we want the model to generate, photorealistic images of our structures. And so how do we go about doing this? So we borrow a lot from the computer vision literature and computer vision and statistical modeling literature. And our first attempt at this is with a computational tool called an autoencoder. In the computer vision literature, often images of numbers for character recognition are put in, and with this you can compress this into a low-dimensional representation, and then from this low-dimensional representation the image can be regenerated. So this is sort of like principal component analysis, if you've heard of that sort of tool. So what can we do with this model with images of our cells? Well, in its sort of vanilla form, this compressed representation is pretty useful. But if we constrain this representation to be sort of normally distributed, such that all of our observations sort of fit to a Gaussian, we can use this as a sort of a probabilistic tool for understanding the cells that we are imaging. If you train one of these autoencoders, you can use this tool to determine how probable the observation that the image of the cell you have is. So you can take this image, pass it into the autoencoder, get the representation in its compressed space, and look up where this number would fall as a draw from a Gaussian distributed variable. Additionally, we can use this as a data generating tool. So because this is a random variable, we can sample from this distribution and then use the autoencoder to generate representative images of our population. And this is particularly useful for exploring variation in the images that we've been acquiring. So let's say I take one of these autoencoders and train it on images of mitochondrial cells, so cells with mitochondria labeled inside of them. And let's say I wanted to see how this population varies. So I can sort of grid sample across this hidden space and generate images of cells that correspond to each location in this low-dimensional manifold. So each of these cells is a synthetically an image of a synthetic cell, so nobody's ever seen these cells before, but they are representative of our population. And because this is a probabilistic model, we can say things like the cell and nuclear shapes in this sort of center area are more common than the sort of cells if you move radially out over here. And this model captures sort of interesting things about the functionality of the structures inside of the cell, such as the cells over here are undergoing mitosis. And those being less common than this cell sort of makes sense because mitosis is a relatively rare event. So this is good, but what we'd really like to do is build models conditional on the images that we've seen before. So in other words, given this image of a cell, can I predict where many, many, many different structures would localize in this cell in particular? So we can expand sort of our autoencoder a little bit and use it to train a model of just cell and nuclear shape, and then build another autoencoder on top of that with the 3-color image with the containing the structure of interest, and have that model learn 3 things. So the first thing is the representation of cell and nuclear shape. The second part that it learns is the labeled structure inside of the image. So you can think of this as an image classifier. And the third part down here is all the other variation that's required to generate this image. So this turns into a model with 3 random variables, 1 random variable that encodes cell and nuclear shape, the other random variable that encodes the structure of interest, and a 3rd random variable that encodes all of the variation in that particular structure. And so with these models, they allow us to build our idea of the integrated cell. So on the left hand here, I'm showing an actual image of a cell and nuclear shape, and holding this constant, I'm predicting the localization of these various different structures inside of the cell. And you can see that the model's learning conditional relationships. So some of these structures localize purely inside of the nucleus, whereas other structures localize in the cytoplasm. And even up here, the cell's about to undergo mitosis. Its chromatin is condensing. And the model predicts 2 mitotic spindles for the alpha tubulin. So so far, we think this is working, but there's some more advancements that we're building into this model. So we've talked a lot about how we build up these libraries of cells and take these images and model them, but perhaps most importantly is how we communicate these advancements to the rest of the world. So with that, we're building a collection of, we're building a website and a collection of tools called the Allen Cell Explorer, which will contain all of the information about our Allen Institute pipeline, as well as the different functional subunits of our institute. In addition to that, we'll have a collection of databases, including the collection of cells that we have, the protocols, tools, and software that we're building here. And we're also building the cell browser that allows that reduces the barrier of entry to accessing our images, and has sort of a plugin-less 3D cell browser, which I think we may demo at one of the poster sessions later. So as a result, our pipeline has several products, starting with the cell lines from gene editing, which are available on the Coriell website. SOPs for that editing, quality control results, as well as cell line characterization and segmentation protocols, raw images and segmentations and features from microscopy, as well as a microscope standardization SOPs. From all of that, we're building predictive models of how structures localize and eventually how cells will behave in the future. And all of that is going to be wrapped together in our Allen Cell Explorer site. So I would like to thank everybody here, my co-presenters, and the rest of the Allen Cell Institute team, and as well as Paul Allen for making all of this possible. So thank you very much. And
so you made a choice to go with microscopy and with a particular kind of microscope. Can you quickly address like what you feel you gained and maybe lost by not choosing other modalities or fixing on a single modality and that one in particular?
Yeah, so one thing that I'll start off with saying is we have spinning disk systems as kind of our system of choice for our first pipeline. We also do have 2 or 3 point scanners as well. So we're definitely interested in exploring other modes of imaging. The reason that we went with spinning disk was it's easy to acquire images quickly, so we can do time-lapse imaging, as well as giving us diffraction-limited high-resolution imaging.
Okay, I think his name was Brock. He talked about damaged brain cells, and
Raise your hand. I can't. Okay,
right here. And would that be like a head injury? And I'm wondering, I'm brain injured and I'm paralyzed on the right side of my body, and I'm wondering, can't you go up in my brain and repair my damaged brain cells? I'm not damaged in the spinal cord. There's no breakage. So it's all up in my brain that I'm paralyzed. I'm here for to get information for the Brain Injury Alliance of Washington, and I'm going to give it to my Dr. Beck at Group Health Hospital. And do you think you could help her to help me?
Yeah, that's a very profound question, and very important. I think it's far outside of where we're working right now, and it's a better question for members of the Brain Institute than for the Cell Institute, I would say. This idea of addressing disease and conditions with induced pluripotent stem cell technology is really, at this point, focused on ameliorating genetic disease rather than injury through trauma. But there's a lot of ideas out there. Normal stem cells. And one possibility for their application is to direct these cells to become neurons and study neurons. I think that we just need to work hard and push this as far as we can in as many different directions as we can. We have to start at the basic level. We're very far from working on brain in a mature sense.
Quick question on modeling. The modeling prediction has, the way you presented it, is going to create a conditional probability on the nucleus and membrane. Do you plan in the future to have conditional relations between many of the individual elements which you are going to measure?
Yes. These images are sort of the first images through our pipeline with just 3 labeled structures. And what we'd like to be able to do is build sort of spatial interaction networks of many of these structures, as well as not only conditioning on different structures, but also conditioning on different experimental conditions. and ultimately allow us to predict spatial relationships between structures, but also predict how those vary as a result of differentiation or some other variable in the experiment.
Do you have any plans to take this from probabilistic space into physical space where you have ODEs or PDEs and you deal with the forces and with diffusion and other physical processes?
The answer is also yes. So right now, the modeling group is just me right now. But we have hired a director, and we're working on building sort of more of a team, a larger team to do that. And so I'm starting with sort of the top-down approach. It's like extraordinarily data-driven, but we certainly intend to use sort of biophysical models and biochemical models to sort of attack this problem from many directions, and as well as use approaches to tie different modeling methods together to get a more coherent view of how cells behave.
It will definitely be multi-scale modeling.
Okay, question here on this side. Hello, here.
Okay.
Okay, hi. So taking cues from the previous two questions, so in terms of the dynamics of the sense, you showed a lot of time-lapse. So I understand it is a complicated problem in terms of deriving the structural models of the entire cell. But then could you comment on what may be potential approaches to incorporate the dynamics, like the temporal dynamics in terms of getting the spatial organization? Because presumably this model that you have developed is from a lot of static time points from a large number of cells.
Yeah.
I mean,
so there's several approaches to do that, and one of the approaches is to sort of encode into the model the position of the cell in the cell cycle, or the position of the cell after the beginning in sort of a dimension that is the time after experiment started. And there's also these same types of probabilistic and image generation models that allow us to input time series, so sort of traces of single cells that allow us to digest and synthesize time series data. So what we'd ultimately like to be able to do once we have some long time series movies is be able to sort of expose the dials of the experiment and be able to say, okay, what does a cell, given the cell now, what would I expect it to look like one time step in the future, and sort of spin this dial and watch the cell move into the future or move back into the past, and things like that. Yes.
Yeah, so given the question about the different components and looking at the relationships between components, is there any plan to multiplex reporters together, so you have multiple reporters in one cell?
Yeah, so the way you could think of these images as having three reporters: the nucleus, the membrane, and the protein of interest. But this sort of ties it back to the spatial interaction question. So we'd like to sort of keep we'd like to vary the reference structures, or vary the pairs or triplets of structures that we observe, and just use those to build these interaction networks.
Yeah,
and Brock says we are creating dual edited lines as well, so we don't have to use dyes.
Tell me, right now you assume that these are all independent instances of cells.
Yes.
And do you know, are these pure colonies, and are they always monolayers?
I'm going to hand that off to some of the people who do cell culture.
Yeah, so actually in collaboration with the stem cell and gene editing group, the assay development team has looked at careful characterization of how the colonies look, both when they're plated in their normal culture state for maintenance and also when we plate them on glass to image them, because people don't typically use these kinds of cells for cell biology. So just establishing what's normal for them was kind of a project. And so yes, we see them as they're epithelial-like monolayers, and they exhibit all the properties you would expect for epithelial cells. So when we look at tagged actin, for example, we see actin bands. We see junctions being formed, adherence junctions with ZO1. We see desmosomes. We see, in some of the images that I showed you with tubulin, we see a primary cilia. So we think they are epithelial cells. They don't form multi-layer structures until they start to differentiate.
So I have maybe a kind of a related question about, I'm over here, hi, about in vivo relevance. So how much of this structure do you think is thought to be a cell culture artifact, as some biologists might put it, versus what you would actually see in an animal? So one example would be to maybe look at structures in the developing blastocyst for pluripotent stem cells and see how those structures, what type of distributions they have versus what you see.
Yeah, we're definitely interested in comparing organization in a cell that's in culture on glass, for example, as compared to part of an organoid.
We're going to start imaging organoids, and that'll bring us closer to the in vivo situation. Not completely, but definitely getting there.